Today we are diving into Chapter 55, Hemostasis and Thrombosis. And honestly, looking at the complexity of this text, it makes the space shuttle look like a Lego set.
It's a beast of a topic. We usually think of blood as just this, this red passive liquid, right?
Yeah.
Like it's just a delivery truck for oxygen.
He's floating around.
Yeah. But this source material paints a completely different picture. It's a picture of blood as a surveillance system. It's an active, intelligent organ that is constantly patrolling your vessel walls, just waiting for a breach.
And that's the tension we're going to unpack today, this delicate balance. We're going to look at how the body pulls off this magic trick, keeping you liquid, but ready to solidify in a heartbeat.
Literally in a heartbeat.
We're going to break down the famous coagulation cascade, which I admit usually looks like a subway map drawn by a lunatic.
We will demystify the Roman numerals, I promise. It's actually much more logical than it looks at first glance. It's just the naming convention is a little unfortunate.
Well, good. We're also going to get into the pharmacology. How do everyday drugs like aspirin or something more serious like warfarin actually hijack this system?
And we have to talk about the history, the royal disease, hemophilia that plagued the monarchies of Europe.
It's a perfect example of what happens when just one tiny gear in this massive, complex machine goes missing. It didn't just affect families, it literally changed the course of history.
It really did.
So let's get into it, the architecture. The text draws a very clear line in the sand right at the beginning between two words that I think most of us, myself included, use interchangeably: hemostasis and thrombosis.
A lot of people do. And in a way, they're two sides of the same coin. But biologically, there's a really important nuance. Hemostasis is the hero of the story.
Okay.
You cut your finger while chopping onions, the blood clots, the bleeding stops. That's hemostasis. It's the normal, physiological, life-saving response to an external injury.
And thrombosis is the villain.
Thrombosis is hemostasis in the wrong place at the wrong time. It's pathology. It's a clot forming inside an intact blood vessel, or maybe a vessel that's just a little bit diseased from, say, atherosclerosis.
So not in response to a cut on the outside.
Exactly. It's an internal event. If that happens in a deep vein in your leg, it's a DVT. If it happens in a coronary artery, it's a heart attack. If it happens in your brain, it's a stroke.
But the mechanism, the actual bricks and mortar the body uses to build the clot, is the same.
It's identical. Whether it's saving your life or ending it, the body uses the exact same three-phase process.
Let's walk through those phases, because the source material implies they happen almost simultaneously in a blur. But for the sake of our sanity, we need to linearize them. So, phase 1, primary hemostasis.
I like to call this the sandbag phase.
The sandbags being the platelets.
Exactly. The very first thing that happens when there's a breach in the vessel wall is that platelets, which are these tiny little cell fragments floating in your blood, get the signal. They rush to the scene.
And just physically pile on top of the hole.
They literally pile up to physically plug the hole. But just like a pile of sandbags in a flood, it's a temporary fix. It's not structurally sound.
Yeah.
If the blood pressure is high, it'll just wash that plug right away.
Okay, so it's a loose plug. You need cement.
That is phase 2, secondary hemostasis. This is where the body weaves an intricate net of a protein called fibrin. This net is the cement. It snakes through the platelet sandbags, catches them, binds them all together, and turns that loose pile into a solid, stable seal.
A proper concrete plug.
A proper plug. And then, interestingly, the text emphasizes a third phase, which I feel like we often forget about in the story of clotting, fibrinolysis. It's the cleanup crew. Because you can't just leave a concrete wall in the middle of your blood vessel forever. That would be just as bad as the leak.
Right, the pipe would be permanently blocked.
So once the underlying tissue is healed, you have to dissolve the clot and restore normal blood flow. If you don't, you end up with permanent blockages.
So logically it goes, build the wall, cement the wall, tear down the wall.
Precisely. A continuous cycle of construction and demolition, all happening on a microscopic scale.
Before we zoom in on the platelets themselves, there was a visual detail in section 1 that I thought was fascinating. The colors of clots. The text differentiates between a white thrombus and a red thrombus. I just assumed a clot was, well, red.
It depends entirely on the physics of the environment where it forms. And this is really, really important for doctors to understand because it dictates how we treat them.
Okay, so take us into an artery. High pressure, fast blood flow.
Okay, imagine a rushing, powerful river. That's your artery. The flow is so rapid that if a clot tries to form, The heavier, bulkier elements, mostly the red blood cells, they just get washed downstream. They can't get a foothold.
They're too heavy to stick.
They're too heavy, and the current is too strong. So what's left behind? Just the really sticky stuff. The platelets and the fibrin strands that form the initial scaffold.
So it's mostly the glue, not the bricks.
An excellent way to put it. So if you were to cut open an artery and look at a clot, say, the kind that causes a heart attack, it looks pale, grayish white. It's a white thrombus.
Okay, now contrast that with a vein.
Veins are the lazy river. Low pressure, slow, sluggish flow. Sometimes, like during a long plane flight or if someone is bedridden, the blood barely moves at all. It's a state called stasis.
And in that environment?
In that environment, everything gets trapped in the fibrin net. The red blood cells, which are huge and incredibly plentiful, get stuck in there along with everything else. So the clot looks deep red, almost like jelly.
A red thrombus.
Right. And here's the kicker, the clinical connection. Because white thrombi are mostly platelets, we treat them with antiplatelet drugs, like aspirin, like Plavix.
Things that stop platelets from sticking.
Exactly. But red thrombi are mostly a fibrin mesh trapping red cells. Platelets are a smaller part of the story, so we treat them with anticoagulants drugs that stop the formation of the fibrin mesh itself.
Like heparin or warfarin?
Precisely. The physics dictates the pharmacology. It's all connected. You can't treat a venous clot effectively with just aspirin, and you can't treat an arterial clot effectively with just warfarin. You have to know the architecture of what you're fighting.
That is very cool. Okay, let's go to section 2. The first responders. The platelets. The text describes them as the sleeping guard.
It's a great metaphor. In a healthy person, you have billions upon billions of platelets circulating at all times. They're shaped like little discs, smooth, flat, completely unassuming.
And they're designed not to be sticky.
They are chemically designed not to stick to anything. They bounce off the walls of your blood vessels like bumper cars. They bounce off each other. They're completely inert.
Because the walls of the blood vessels are lined with this special layer, the endothelium.
Correct. The endothelium is the Teflon of the body. It's an incredibly sophisticated surface. It doesn't just provide a physical barrier, it actively repels clotting. It secretes chemicals like nitric oxide and prostacyclin that send a constant signal to the platelets saying, keep moving, nothing to see here, stay calm.
Until the Teflon gets scratched.
Right. If you cut the vessel, or, more commonly, if an atherosclerotic plaque inside an artery ruptures, you strip away that protective endothelial lining. And what's revealed underneath it is a layer of protein called collagen.
And collagen is the trigger.
Collagen is the universal alarm bell for platelets. Platelets have specific receptors on their surface that are, in essence, always sniffing for collagen. As soon as they touch it, they undergo a transformation that is honestly terrifying if you look at it under an electron microscope.
Describe it.
They stop being these smooth little discs. They explode. They become these spiky spheres. They shoot out these long, tentacle-like arms called pseudopods. They effectively become shapeshifters, turning from a smooth stone into a sticky burr.
And those arms, the pseudopods, what are they for?
They're for grabbing. They use these arms to reach out and grab onto the exposed collagen, anchoring themselves against the flow of blood. And they also use them to grab onto each other to start building that initial sandbag wall.
And this isn't just a physical change, right? There is some heavy-duty computing happening inside the cell. The text talks about inside-out and outside-in signaling. That sounds complex.
Yeah, this is where the biochemistry gets really elegant. So think of the platelet as having a sensor on the outside. That's the outside-in part. It touches collagen or it gets hit by a chemical messenger like thrombin.
So an external signal.
An external signal. That mechanical touch or chemical binding sends a signal into the center of the platelet. It's a chain reaction.
And what's the ultimate message of that chain reaction?
The message is DUMP, the calcium.
It always comes back to calcium in biology, doesn't it?
It really does. The text details this whole pathway. Phospholipase C beta hydrolyzes a lipid called PIP2 into two smaller molecules, DIE and IP3. You might remember those.
Vaguely, they're second messengers, right?
Exactly. And IP3's only job is to go to these internal storage granules, these little sacs of calcium, and unlock them. So the concentration of calcium inside the cell just spikes 1,000-fold in a split second.
And that calcium spike is what causes all the changes.
It's the master switch. That flood of calcium causes the platelet to change its shape, and it also causes the platelet to vomit, is the word the text almost uses, it degranulate.
Vomits is a visceral word.
It's accurate. It has these storage pods full of chemicals, and it just dumps its entire payload into the surrounding blood. Chemicals like ADP and serotonin, these are signals to call for help.
And this leads to the inside-out part of the signal.
Exactly. That same internal calcium spike also sends a signal back out to the surface. It changes the shape of other receptors on the outside of the platelet, specifically a very important one called GPII-IIIA.
Okay, I'm just going to call that the Velcro receptor.
That works perfectly. It is the Velcro. Normally, this receptor is a fold and shut. It's inactive, but when that wave of calcium hits it from the inside, it undergoes a conformational change. It springs open.
And when it's open,
It becomes incredibly sticky. And what it's designed to grab is a protein that's always floating around in the blood called fibrinogen. So it grabs one end of a fibrinogen molecule, and another platelet grabs the other end.
so it links them together.
It's how the platelets chain themselves together to form a solid mass. So the signal goes in to wake the cell up, and the cell then sends a signal out to deploy its grappling hooks. It's Velcro.
Perfect summary. But there's a physics problem we need to solve here. You mentioned earlier that arteries are like rushing rivers.
Right, high shear stress. The force of the blood flow is immense.
So if you're a tiny platelet trying to grab onto a piece of collagen in what is essentially a class 5 rapid, you're just going to get ripped away, right? Your grip isn't strong enough.
That's a huge problem for the body to solve. And it has an incredibly clever solution. It's a protein called von Willebrand factor, or VWF.
Okay, what does that do?
Von Willebrand factor is a massive, long, stringy protein. Think of it like a piece of molecular bungee cord. One end of it anchors deep into the collagen of the damaged vessel wall.
So it's got a solid anchor point.
A very solid anchor. The other end uncoils into the bloodstream and acts like a lasso. It has a specific binding site that grabs onto a receptor on the platelet.
It is the bridge.
It is the bridge that tethers the platelet to the wall, allowing it to adhere even when the blood is rushing by at high speed. Without VWF, you simply cannot form a stable platelet plug in arteries.
Which is exactly the problem in von Willebrand disease, which we'll touch on later. But for now, the platelet is stuck, it's activated, and you said it's dumping chemicals, specifically ADP and thromboxane A2.
Right, this is the call for backup phase. The first platelet to arrive doesn't want to work alone. It releases ADP from its dense granules to wake up all the other sleeping platelets flowing past. It's basically shouting, "Hey, party over here, pile on."
And thromboxane A2, what does that do?
Thromboxane A2 is the heavy artillery. It's synthesized fresh inside the activated platelet from a fatty acid called arachidonic acid. It does two critical things. One, it's an incredibly potent activator of other platelets, making them super sticky. And two, it's a powerful vasoconstrictor.
Meaning it squeezes the blood vessel shut.
It causes the muscle in the vessel wall to clamp down, which physically reduces the size of the hole and limits blood loss. It's a brilliant 2-for-1 molecule.
Okay, so thromboxane A2 is a potent promoter of clotting, which brings us directly and perfectly to Section 3, pharmacology. Because if you have a patient at risk of a heart attack, which is an unwanted clot in the heart, you desperately want to shut this system down.
And this is the absolute genius of a simple 100-year-old drug, aspirin.
I was surprised by how specific the mechanism is. I mean, you take an aspirin for a headache and you don't think twice, but chemically, it is doing something remarkably ruthless and specific.
It's pure chemical sabotage. So to make that thromboxane A2 molecule, the platelet needs a specific enzyme, a little biological machine. It's called cyclooxygenase 1, or COX1.
Okay.
Aspirin gets absorbed into your bloodstream, it enters the platelet, and it finds that COX1 enzyme. It goes to the active site of the enzyme, and it performs a chemical reaction called acetylation.
Acetylation, what does that mean in simple terms?
It attaches a small chemical group that physically blocks the enzyme's machinery. It jams the gears. And here's the crucial part that makes aspirin so special. It is an irreversible jam. It breaks the enzyme permanently.
But in most of our tissues, if you break an enzyme, the cell just makes a new one, right? The nucleus has the DNA blueprint.
Absolutely. If this happens in, say, a cell lining your stomach, that cell just reads the blueprint and builds a new COX1 machine in a few hours. No big deal. But remember, platelets are not true cells.
Right, they're just fragments.
They're fragments of a larger cell. They have no nucleus, they have no DNA, they have no blueprints.
They're no factory to build new parts.
Exactly. So when aspirin breaks their COX1 enzyme, that platelet is permanently disabled. It can never make thromboxane A2 again for the rest of its life.
Which is why the effect of a single aspirin lasts for about 7 to 10 days. The entire lifespan of that platelet.
Precisely. Even after the aspirin is long gone from your system, the effect on your blood stickiness remains until your bone marrow produces a whole new batch of fresh platelets.
And that's why we can use such a tiny dose, a baby aspirin, 81 milligrams, You don't need a huge amount to suppress the whole body. You just need enough to knock out the circulating platelet pool.
It's an incredibly elegant mechanism.
Yeah.
And the text also points out a fascinating balance. The endothelial cells, the Teflon lining, they also use a COX enzyme to make prostacyclin, which is an anti-clotting molecule.
So it's the good guy.
It's the good guy. Aspirin blocks that too, which is bad. But because endothelial cells have a nucleus, they can regenerate their COX enzyme in a few hours. The platelets can't. So a low dose of aspirin tips the balance in favor of anti-clotting.
That's amazing. And what about other antiplatelet drugs like clopidogrel or Plavix?
Plavix works on the other signal we discussed, ADP. It blocks one of the key ADP receptors, the PDY12 receptor. So the first platelet can still shout, "Help!" by releasing ADP, but the other platelets have earplugs in. They don't hear the signal, so they don't get activated, and they don't cluster.
So for a really high-risk patient, someone who just had a heart attack or a stent put in, you might give them both.
That's called dual antiplatelet therapy, or DAPT. You hit them with aspirin to block the thromboxane pathway, and you hit them with Plavix to block the ADP pathway. You're essentially dismantling the two major communication networks that platelets use to aggregate.
It's a two-pronged attack.
A very effective one. And the text mentions a third class, drugs like ipsiximab, which directly block that Velcro receptor, GPIIIE. These are incredibly potent, usually given IV in the hospital for high-risk procedures. They just stop the platelets from linking together at all.
Okay, so we have a pile of platelets. They are stuck together, but as you said, they're just sandbags. Now we need the concrete, we need fibrin. And to get fibrin, we have to navigate the waterfall.
The coagulation cascade.
I remember looking at a chart of this in college and just my eyes glazed over. It's a soup of Roman numerals. Factor 12 activates X11, which activates NIX, which combines with 8. It felt completely random.
It feels random because of the naming system. The numbers, factor 7, factor 8, factor X, factor NIX, they were assigned in the order they were discovered by scientists, not the order in which they react in the body.
That explains so much. It's not a sequence.
It is not a numerical sequence at all. If you strip away the numbers, the concept is actually beautifully simple. It's a series of what are called zymogens.
Define zymogen for us.
A zymogen is a soldier standing at attention with his weapon on safety. It's an enzyme, usually a protease that cuts things circulating in your blood, but it's inactive. It's just waiting.
Until it gets an order.
Right. And the order usually comes in the form of being cut itself.
Right.
A previously activated enzyme comes along and snips a small piece off the zymogen. That snip is like flipping the safety off the rifle.
And now it's an active enzyme.
Now it's an active enzyme. We usually denote that by adding a little A to the name, so factor X becomes factor 6A, and its only job is to go find the next zymogen in the line and snip him to activate him.
So it is a domino effect.
It's a domino effect with massive amplification. One activated factor 12 might activate 100 molecules of factor 11. Those 100 factor X's might activate 10,000 molecules of factor IX. By the time you get to the bottom of the waterfall, a single initiating event has resulted in millions of molecules reacting almost simultaneously.
It's an explosion. Now, the text divides this into two traditional roads, the extrinsic pathway and the intrinsic pathway. Why do we need two different roads to get to the same place?
It's all about speed versus sustainability. The extrinsic pathway is the spark. It is incredibly fast.
What triggers it?
It's triggered by something called tissue factor. Tissue factor is a protein that is present on cells outside of the blood vessel. Under normal conditions, your blood never, ever sees tissue factor.
But if you get a cut?
If you get a traumatic injury, a knife cut, a serious wound, the barrier is broken. Blood spills out and comes into contact with tissue factor. And boom, tissue factor grabs onto factor 7 floating in the blood, and that complex immediately activates factor X. It's a very short, very fast pathway.
So it's explosive.
It's explosive, but it kind of burns out fast. It produces a quick, small burst of clotting. It's not enough to build a big, stable, durable clot on its own.
So for that, you need the intrinsic pathway.
The intrinsic pathway is the log fire. It takes a little longer to get going because it involves more steps, the classic sequence of factor 12 to Xella to IX. But once it catches, it burns hot and for a long time. Its main job in the body is to amplify the signal started by the extrinsic pathway.
And this is where hemophilia comes in, right? Because hemophilia affects factors in the intrinsic pathway.
Exactly. This is the crucial point. Hemophilia A is a deficiency of factor 8. Hemophilia B is a deficiency of factor IX. Both of those are key components of the intrinsic pathway's amplification loop.
So walk us through it. If a person with hemophilia gets a cut, what happens?
Well, their extrinsic pathway works perfectly fine. Their blood hits tissue factor, their factor 7 gets activated. They make a little bit of clot. So initially, the bleeding might stop.
Which would give you false hope.
Exactly. But because they're missing the amplifier, they're missing factor 8 or factor 9X, they can't sustain it. They can't build up a big, strong fibrin mesh. The log fire never catches.
The spark just dies out.
The spark dies out. The weak initial clot dissolves under the pressure of the blood, and they start bleeding again, sometimes hours later. That's the tragedy of the disease. It's not the inability to start clotting, it's the inability to maintain the seal.
Now, both of these roads, the fast spark and the slow-burning fire, they eventually merge. They meet at the activation of factor X. This is called the common pathway.
All roads lead to Rome, and in coagulation, Rome is factor X. Once factor X is activated to factor SAC, it doesn't matter how you got there.
And what does factor ZAG do?
Factor XAN teams up with its cofactor, the activated factor VA, on the surface of platelets. Together with calcium and phospholipids, they form a superenzyme complex called the prothrombinase complex.
That sounds like a heavy metal band.
It acts like one. It absolutely shreds. The catalytic efficiency of this complex when it's assembled on a membrane is orders of magnitude, hundreds of thousands of times more powerful than the individual enzymes floating free.
And its only job?
Its only job is to take prothrombin, which is factor 2, and convert it into the absolute king of the cascade. Thrombin, the generation of thrombin, is the whole point of everything we've talked about so far.
The text really puts thrombin on a pedestal. It calls it the master conductor.
As it should. Thrombin is the central figure. It doesn't just do one thing. It's a multifunctional enzyme. First, it goes back and acts as a potent platelet activator, strengthening the sandbag wall.
Okay.
Second, it goes back upstream in the cascade and activates factors V, 8, and 11. This creates a massive positive feedback loop. Thrombin essentially screams more thrombin at the system.
It accelerates its own production.
Massively. And most importantly, of course, it builds the fibrin mesh.
That's section 5, building the mesh. So thrombin finds fibrinogen. Now, fibrinogen, you said, is soluble. It's just floating around in the blood plasma, minding its own business.
Correct. It's a liquid protein. The text describes its structure, a sort of trinodular rod, and it has these little negatively charged caps on it called fibrinopeptides A and B. These caps make the molecules repel each other, which keeps them from clumping together.
So thrombin is the barber.
Thrombin is the barber. It comes along with its molecular scissors, and it snips off those fibrinopeptide caps.
And once the caps are off?
The molecule fundamentally changes. It's no longer repulsive. It develops these sticky ends. It becomes insoluble, and it wants to spontaneously polymerize. It binds to other fibrin monomers that have also had their caps snipped off.
And they just line up on their own?
They naturally line up in this beautiful half-staggered pattern to form long strands called protofibrils. These then associate side to side to form the fibrin fibers that we see as a clot.
But the text says these strands are weak initially. They're like stacking Legos without actually clicking them in.
Right. They're just held together by weak, non-covalent hydrogen bonds. You could pull them apart with a bit of force. To make it a permanent, strong seal, you need one final player. Factor 13.
The baker's dozen.
The baker's dozen. Thrombin's other job is to activate factor 13 to factor 13. Factor 13 is an enzyme called a transglutaminase. It acts like a molecular welding torch.
A welding torch.
It forms powerful covalent chemical bonds between the adjacent fibrin strands. It literally cross-links them together. Once factor 13 has done with its work, that clot is tough. It's mechanically strong, rubbery, and resistant to being broken down. It's not going anywhere.
It's a staggering amount of complexity. But this leads to the obvious question that the text addresses in section 6. If this system is built on positive feedback and amplification, one molecule leads to millions. Why does it not just keep going? Why does a paper cut on my finger not turn my entire bloodstream into a solid block of Jell-O?
That is the regulation problem. It's probably the most important part of the whole story.
Right.
And the answer is that the body puts just as much energy into building the brakes as it does into building the engine.
So let's talk about the main brake. Antithrombin. The name is pretty self-explanatory.
It is. Antithrombin is a protein that's always circulating, always patrolling the blood, looking for rogue active enzymes like thrombin and factor ZA that have drifted away from the site of injury.
And when it finds them?
When it finds them, it binds to them and neutralizes them. It's a suicide inhibitor. It hugs them to death, forming a one-to-one complex that is then cleared from the body. It accounts for about 75% of the anti-clotting activity in the blood.
And this is where the drug heparin comes in. I always associate heparin with hospitals.
Right. This is a really cool molecular mechanism. On its own, antithrombin is, well, it's kind of a lazy cop. It works, but it's slow.
Wow.
Heparin, which is a molecule found on the surface of our own endothelial cells, acts as a catalyst.
It speeds it up.
It speeds it up by over 1,000 times. Heparin binds to antithrombin and causes a conformational change. It changes its shape.
Like popping open a bear trap.
Exactly. It exposes the active site of antithrombin, making it incredibly efficient at grabbing thrombin. That's why if someone comes into the hospital with a big clot, we give them a shot of heparin IV. It instantly supercharges their body's natural braking system.
Wow. And there's another braking system that I found really clever in the text, the protein C pathway, because it actually uses thrombin against itself.
It's a brilliant traitor mechanism.
Right.
So when thrombin strays too far from the injury site and bumps into the healthy, undamaged endothelial cells lining the vessel, it finds a specific receptor waiting for it called thrombomodulin.
Thrombomodulin.
It modulates thrombin. When thrombin is bound and held by thrombomodulin, it undergoes a complete personality change. It stops all of its pro-clotting activities. It can't activate platelets. It can't make fibrin. Instead, it gains a new ability. It activates a zymogen called protein C.
And what does activated protein C do?
Activated protein C, along with its cofactor protein S, becomes a clot buster. It goes back and finds two of the key amplification factors, factor VA and factor eta, and it destroys them. It chops them up.
So it cuts the fuel line to the engine that created the thrombin in the first place.
It's a perfect negative feedback loop. It's a system that says clotting is allowed here at the site of injury, but not out here on the healthy surfaces. It ensures the fire stays in the fireplace and doesn't burn down the whole house.
And this brings us to a term I've heard before, factor V Leiden. I know it's a fairly common genetic condition.
It is. It's the most common inherited risk factor for thrombosis. People with a factor V Leiden mutation have a tiny change, a single amino acid substitution, in their factor V protein.
Does it stop factor V from working?
No, and that's the interesting part. Their factor V works just fine to help make clots. But, and here's the critical problem, that single mutation happens to be in the exact spot where activated protein C is supposed to cut it.
So the brakes don't work on it?
Exactly. Activated protein C comes along to try and shut down the clotting, but its molecular scissors can't cut the mutated factor V. The blade just bounces off. So the clotting engine continues to run for much longer than it should. These people are hypercoagulable. They are prone to developing DVTs and pulmonary embolisms because their built-in braking system is faulty.
It's amazing how a single letter change in your DNA can change the entire risk profile of your life, which is a good transition to Section 7, warfarin and vitamin K.
The rat poison connection.
We have to talk about that. It's such a strange story.
It really is. Warfarin, or Coumadin, was originally discovered because farmers in the 1920s noticed their cattle were mysteriously bleeding to death after eating moldy, sweet clover hay.
That's bizarre.
It took years, but researchers at the University of Wisconsin figured out there was a compound in the mold that was acting as a powerful anticoagulant. They isolated it, called it warfarin, from the Wisconsin Alumni Research Foundation. And its first use was as a very effective rat poison.
Because rats would eat the bait, and then a day or two later they'd bleed out internally?
Grim, but effective. And then in the 1950s, a doctor had the brilliant idea that, hey, in tiny controlled doses, this could save humans who are at risk of strokes from things like atrial fibrillation.
But why vitamin K? What does this vitamin actually do in the cascade?
This is the key to everything. The liver makes several of the key clotting factors, 2, 7, I, X, and X, plus the regulatory proteins C and S. But when they first come off the assembly line, they're duds, they're toothless.
Why? What are they missing?
They're missing the ability to stick to the platelet surface. Remember, all the action happens on the phospholipid membrane of the activated platelet.
Right.
But platelet surfaces are negatively charged, and the clotting factors have regions that are also negatively charged. They repel each other, like trying to push two same-sided magnets together.
So you need a bridge.
You need a calcium bridge.
Mm-hmm.
Positively charged calcium ions, pectate 2+ to wall, act as the glue. But the protein needs a way to hold on to the calcium. And that's where vitamin K comes in. It's the fuel for an enzyme that adds an extra carboxyl group onto specific glutamate residues on the protein.
So it turns a glue into a GLA, as the text says.
Exactly. It creates a gamma-carboxyglutamate, or GLA domain. And that GLA domain is a perfect negatively charged claw for grabbing onto positively charged calcium.
No vitamin K, no GLA, No claw, no calcium, no binding to the platelet.
And no binding to the platelet means no clot.
Mhm.
The whole cascade falls apart. So warfarin works by inhibiting the enzyme that recycles vitamin K in the liver, so you effectively run out of it.
Which is also why warfarin takes days to work, right? You have to wait for the old functional factors that are already in your blood to be used up and die off.
Exactly. It's not an instant effect like heparin. And to reverse it, you can give a big dose of vitamin K, but that's slow. If it's an emergency, you have to give the person an infusion of fresh frozen plasma, which contains all the functional clotting factors they're missing.
Let's wrap up the pathology with Section 8, the royal disease, hemophilia.
It's a fascinating intersection of molecular biology, genetics, and world history. It all seems to trace back to Queen Victoria of England. She was likely a carrier of a spontaneous mutation for hemophilia A on one of her X chromosomes.
She didn't have the disease herself?
No, because it's an X-linked recessive disorder. Women have two X chromosomes, so her other, healthy X chromosome was able to make enough factor VIII to keep her fine. She was just a silent carrier. But she passed that faulty X chromosome to several of her children.
To her son, Leopold, who had the disease, and to several of her daughters who became carriers.
Right. And through the strategic marriages of the 19th century, those daughters spread the gene into the royal houses of Russia, most famously, to the Tsarevich Alexei, and also to the royal families of Spain and Germany.
And clinically, the text emphasizes bleeding into joints. Why the joints? Why do people with hemophilia bleed into their knees and elbows so often?
Think about the mechanics of everyday life. Your joints take a constant beating. Every time you walk or run, you're causing microscopic trauma. Tiny little capillaries break inside the joint capsule.
But in you and me, the system just patches it instantly. We never even notice.
We never notice. A few platelets and a dab of fibrin, and it's fixed in seconds. But for someone with hemophilia, the patch fails. The bleeding continues, slowly but surely, into the closed, confined space of the joint.
And the pressure builds up.
The pressure builds up, the blood irritates the lining of the joint, it causes inflammation, and over time, it destroys the cartilage. It's incredibly painful and crippling.
And what about von Willebrand disease? The text says it's way more common.
It's the most common inherited bleeding disorder by far, affecting up to 1% of the population to some degree. It's a defect in that von Willebrand factor protein we talked about earlier.
It's a molecular bungee cord.
The bungee cord. So people with VWD have two problems. First, their platelets can't stick properly to the vessel wall, especially under high flow. And second, a lesser-known job of VWF is to protect factor VIII from being destroyed too quickly in the bloodstream. So they often have low levels of factor VIII as well. It's like a mild form of hemophilia combined with a platelet defect.
Okay, so finally, Section 9. We've built the clot, we've regulated it. Now, after the wound is healed, we need to destroy it. Fibrinolysis.
The yin to the yang of coagulation. We have this incredibly powerful system to build fibrin, so we absolutely must have an equally powerful system to cut it apart. The main scissor here is an enzyme called plasmin.
And plasmin circulates as an inactive plasminogen?
Yes, and cleverly, plasminogen gets trapped inside the clot as it's being formed. It's built right into the structure, just waiting for an activation signal.
And that signal is the clot buster drug, TPA.
Right. TPA stands for tissue plasminogen activator. It's an enzyme released by our own endothelial cells, especially under stress. Its job is to find that plasminogen that's trapped in the fibrin mesh and activate it to plasmin.
So if someone is having a stroke from a clot in their brain and they get to the ER fast enough, we can give them an injection of recombinant man-made TPA.
Exactly. We give them a huge dose of it. It floods their system, finds the clot, converts the trapped plasminogen to plasmin, and plasmin starts chewing up the fibrin mesh, hopefully dissolving the clot and restoring blood flow to the brain before the tissue dies.
But the coolest story in this section has to be streptokinase.
Oh, the bacterial hacker. It's an amazing story of evolutionary warfare. Streptococcal bacteria, the kind that causes strep throat, evolved a protein called streptokinase.
And why would a bacterium want to make a clot-busting protein?
Well, think about it from the bacteria's point of view. When it invades your body, one of your body's defense mechanisms is to try and trap it. It forms a fibrin clot around the infection to wall it off and stop it from spreading.
So the bacteria says, nice try, and releases streptokinase.
It releases its secret weapon. Streptokinase activates your own plasminogen to dissolve your own defensive clot. This allows the bacteria to escape the quarantine and spread throughout your tissues.
That is biological warfare at its finest.
It is. And for decades, we basically stole their weapon. We harvested streptokinase from bacteria cultures and used it as the very first clotbuster drug to treat heart attacks. We used the bacteria's key to unlock our own clots.
That is a perfect place to bring this all together. The idea that some of our most advanced treatments come from understanding these microscopic wars that are constantly happening inside us and around us.
It's incredibly humbling. The bacteria figured out the principles of therapeutic fibrinolysis millions of years before we did.
Let's just do a quick run-through of the labs in Section 10 before we sign off, just so our listeners might recognize what to look for on a blood test result.
Sure. If you want to get a snapshot of how this whole system is working, there are a few key tests. First and simplest is the platelet count. Do you have enough sandbags?
Simple enough.
Second is the APTT, or activated partial thromboplastin time. This test measures the integrity of the intrinsic pathway, the log fire, factors 12, LAX, 88. This is the test we use to monitor someone on heparin therapy.
And the third?
The PT, or prothrombin time. This measures the extrinsic pathway, the spark, mainly factor 7. This is the test, often reported as the INR, that we use to monitor patients on warfarin.
There was a mnemonic you mentioned to me earlier that I thought was great.
Oh, right. Play table tennis, or PTT, is played indoors, so it measures the intrinsic pathway. Paratroopers, or PT, come from the outside, so it measures the extrinsic pathway.
I love that. It makes it stick. So what is the single biggest takeaway for you from this deep dive into Chapter 55?
For me, it's the sheer resilience of the system. We have these explosive positive feedback loops that could kill us in minutes, and we have these incredibly powerful negative feedback loops that can make us bleed out. But 99.999% of the time, the balance holds. You stay liquid, but you don't leak. It's this dynamic, precarious equilibrium that requires millions of microscopic adjustments every single second of your life.
And next time you get a paper cut, maybe give a little nod of respect to the platelets and the fibrin mesh and factor third wick overtime to keep you intact.
They are absolutely the hardest working, most underappreciated cells in the business.
This was fantastic. Thanks for diving deep with us.
Always a pleasure.
Stay curious, and we'll see you on the next one.